Now the fun begins. 11.5 starts a section called Non-Mendelian Genetics. So everything up until this point, up until 11.4, we've really been talking about things called Mendelian genetics. Mendelian genetics are very easy. They follow very simple rules, and if we talk about Mendel and how he chose the pea plant, he actually got kind of lucky. Pea plants really only have one of two phenotypes, and they're really easy with genetically to observe and to get data from. The the reality is genetics don't really follow those rules all the time. They will follow some of those rules, but then they have exceptions to those rules, and we're going to see a lot of exceptions to those rules today. I always so my I talk about my best friend that's a doctor. He was a geneticist, and he is going to be a doctor of geneticist, if that makes sense. And he always talks about how there is just so hard to really know what you know in genetics. So whenever somebody says like, "Oh, this is the way," and it's like, well, it actually doesn't really. When you dive deep into something, it doesn't really work that way. So we're going to see complexity increasing here and a lot of exceptions to those rules. It doesn't disprove Mendelian genetics. It just says, hey, there's a lot more stuff out there. Now, with all that being said, I'm going to get to the actual material here. Multiple allele traits. I don't have a worksheet for this. I do, but I don't include it into my packets. But this is just that there are more than there are more than two alleles. You can see the big B, little b here. There are more than two alleles here that contribute to the phenotype of an organism. In this case, you can see the big C, the little c with the CH, the little c with the H, and the little c. And they all have different phenotypes. So you can see there are multiple alleles. And this is just where there's four different alleles. There are going to be some that have many more than just four different alleles, so that it's not just like purple or white flowers, tall or short pea plants. It's more than two, and this is the most often the case is that there's just more of them. You can see most of human genetics are multiple allele traits. The HLA system. This is what tells your body if like a cell is your cell or not. There's over 30 million different types of genotypes for this. There are a ton of different genotypes for the HLA system. Codominance. So moving right along, codominance is where there are several alleles that are equally dominant to each other. So you can see here we have this. It's called the roan cow down here. We have pure red cows, pure white cows that are both signifying with like a capital letter. And when you mix them, you get this kind of splotchy. It's again because the genes in the R category here, the R allele, are being expressed in the R fur, and the white genes are being expressed in the white fur. Now, the human blood type actually follows both of these multiple alleles in codominance. A and B are both dominant to O, which is going to give us four different phenotypes. We have A, we have B, we have AB, and we have O. Now, with these alleles, we can have an A and an O because A is dominant to O, but it's still going to produce A blood type. We also have A and A. We have B and O, which produces B blood type, but we also have BB. AB blood type only has one genotype, which is AB. And we have O blood type, which is created by two O alleles, or they're signified by little i's here. This just shows you that the I capital A is dominant to the I, which is the O blood type. So again, you can see here the antigens or the human blood type alleles. Now, what they actually do is the A antigen creates something called, I'm sorry, the A allele creates something called the A antigen. You can see how these antigens look different than the B antigens. And then the AB group, they both produce A and B antigens. The O group doesn't have any antigens that the A and B have. The people with A blood also create something called B antibodies, and these B antibodies will attack B blood. The people with B blood will create these A antibodies that attack people with A blood or attack the blood of A. And then AB people don't have any antibodies, so they are going to be our universal receivers because they don't have any antibodies to attack blood with. O blood has both A and B antibodies, which attack both A, B, and AB blood. However, these guys don't have any antigens on them, so they can be given to anybody. So being A, B, and O is kind of cool because, again, AB can receive from anybody, O can give to anybody, and yeah. we can see here what's going to happen is these red blood cells are going to be attacked by these antibodies. These antibodies are going to coagulate the blood and cause major problems and actually can kill the person if they. So, like if I have A blood, if I get B blood, I could have serious health concerns or maybe even die because of this coagulation and my antibody response. So I'm not going to go through all of these. I will go through them in class, but just understand that you'll have to do these Punnett squares with the ABO blood system, and you get a lot of practice with the packet. Type O blood is a universal donor, like I said, because they have no antigens. Type AB blood is a universal receiver because again, they don't have any antibodies and they have both A and B antigens on their blood. So you can see the donor chart here. O can give to anybody, AB can get to anybody, AB can only give to AB, B can give to B and AB. That makes sense. You can just kind of figure out this chart. Now, we also have these things called positive and negatives, and these positive and negatives refer to the RH factor. The RH stands for the rhesus monkey, who we have a very common ancestor with about, I think, like 10 million years ago or something like that. But these rhesus monkeys, and again, the reason why they actually call it the RH factor is they first found these in rhesus monkeys, and then they found them in us, and they're like, oh, they're the same antigens. That's pretty cool. So if you are positive, that means you are either homozygous or heterozygous for positive, which means you have these antigens. If you're negative, you don't have these antigens, and you're again homozygous for this as well. So the recessive here is the negative. So you can see here these Rh factors with the positive. They're not here for the negative. So the blood types, again, we can have AB positive, which has the most unique antigens on their blood cells. The AB negative has, again, just A and B. Then the A, B negative, O negative. They don't have any of these negative rhesus factors that that the positive people have. So, like I said with the beginning of this section, non-Mendelian genetics aren't going to follow all the rules that Mendel saw in his pea plants, and this is one of those cases. Now, before you even kind of like we dive down deep into this, don't automatically think, oh, this is the blending hypothesis. It's not the blending hypothesis. So if we look here, we have these heterozygous. They're going to be kind of a mix between the red and the white. So we can see here there's an intermediate color. And it again, it does look like the blending hypothesis. And this is why some scientists before the 1800s really thought, hey, we see things mixing in nature because it sometimes does. So here's the thing. This isn't the blending hypothesis because if we still mix these pink flowers, so we get two pink flowers here, we will get some pink flowers, but we will also get red flowers and white flowers. So this proves the blending hypothesis still. So don't think this is the blending hypothesis. You can see it's wrote usually as a superscript, and we will do a page in your packet on the incomplete dominance. Sex-linked genetics. Any gene that is located on a sex chromosome. So any gene where we have it on the X chromosome is labeled as a sex-linked gene. This is where we're going to see males having diseases and disorders more often than females, because we only have one X chromosome, and females have two, which means they have to get two of these X chromosomes, which is much, much, much more unlikely than a guy getting one. So again, males only need one copy. Let me go back here. Males only need one copy of this disorder, and you can see here it's usually recessive. It can be dominant, but they're usually more recessive. Females need two. So you can see here this disorder they need two. We have the again. I'm just going to move on from this. We don't need to see this again. Color blindness is an example of one of these X-linked disorders where, again, it's in 1 in 12 men. So we see this a lot more in men than females. It's about 1 in 200 females. Again, it's on the X chromosome. So a guy with one X chromosome with this disorder will automatically have it. A female that has just one copy will be a carrier. So she'll still see in color, but oddly enough, remember, we're going to talk about how both of these genes are expressed. She won't see as many colors as somebody with two X chromosomes that have a quote unquote normal or unaffected gene. Now, I'm going to briefly show you some color, like what it would look like if you had like color deficiencies. Now, don't freak out. Again, with the test here, if you can't read this, the color doesn't always come through with these pictures. Go see a doctor if you do think you're colorblind a little bit. This is 74. I can sometimes see how somebody would say 71. You can see here the 71. So this helps them or doctors identify if you're colorblind or not. I'll take you through some of these X-linked disorders in class, but I'm not going to do it here. Y-linked disorders, yep, we do have very, very few, and they're again, they're rare on the Y chromosome. And again, this is only when we see males getting it and passing it on. So you can see here, only the males are going to pass it on. And again, these are very rare. Our next genetics that we're going to be talking about within non-Mendelian genetics is polygenic inheritance. This is one of my favorites because it really gets to show you that not all of our genetics plays by Mendel's laws, where it's only one of two phenotypes. In this case here, we can see there are many different phenotypes just within, I think this is like a 1993 Connecticut class or whatever, and they did this. I'd love to do this with our students one day, but obviously with COVID, we're not going to be able to do it. You can see here, there are a lot of different phenotypes. And if we made this like 5 foot 1/2, 5 foot a quarter, there would be even more phenotypes. But what you can see here is there's this bell curve. So a lot of times with these polygenic inheritance, there are several genes that are going to contribute to a single phenotype. Eye color is one of them, height's another one. And again, most of human genetics are going to follow this, where you have this bell curve of phenotypes. For example, here, and this is not saying this graph's very interesting. I love using this graph because it gets to show you that it's not that females are taller than men, because that's obviously not true. Men are taller, but it shows you that the females have a kind of shorter standard deviation and have a wider standard deviation, and they are taller. You can see heights on the bottom here and the frequencies up top. So we see this a lot of time in men. Men have a little bit longer, a little bit wider standard deviation. There's just more genetic variation in the guys we see that deals with evolution and things like that, which we'll get to later on. I like to show this because this shows you like the tall gene. A capital letter is a growth hormone, and so the lowercase letter is the absence of that, or like a protein that maybe doesn't help the person grow. We can see here that if we have two people that are heterozygous, most of the time you can see these middle ones are going to have three. Have these capital letters. That means they're going to be average height. So these people are average height. Most of their kids are going to be average height. Is it possible that they get a really, really tall kid? Yeah. Is it possible to get a really, really short kid? Yeah, but they're very, very rare. So this is like a person that is, again, average height, having kids. If we have two of these people having kids, they're probably going to have a lot of tall kids. If we have two of these people having kids, they're probably going to have a lot of short kids. So this is just like rolling the dice on how many of these you get. Eye color is another one where it's not just dominant recessive and you have two genes. There are many genes that affect eye color. So we usually don't use these in the Mendelian portion because it's not correct. It's there's actually a big push in genetic education that we say, hey, don't use these in your regular Mendelian worksheets because they don't follow those rules. I'll talk about this a little bit more in class, but there are certain percentages you can guess. So I've also had said, had students say, "Hey, Mr. Frath, both my parents have blue eyes, but I have, green or brown eyes. Does that mean something happened?" No, there is a chance you could get brown eyes from having two blue eye parents. Very rare, but there's still a chance. So I always say, don't freak out. Like, don't think like you're adopted or something like that, and go. There's a lot of genetic variation that goes on here, and it's tough. Don't automatically think because you see something here. It's very tough to talk about these things and have absolutes. Skin color is another one where we have again this pigment that's going to be created with these capital letters. Extremely light people are going to have the lack of that pigment, and I'm going to show a video in class that really shows you why evolutionarily we have different skin colors, and it's very interesting. It's very cool, and the research was actually done at Penn State University, which is just right down the road. It's really cool. Oh, I'm not going to show that video. I think this is it. Oh no, we have a couple other ones. Incomplete penetrance is when you do get a genotype, but you won't produce the phenotype. So just because you get, it's called the BRCA1 gene, just because you get this BRCA1 gene mutated, it doesn't mean you always will have breast cancer. This BRCA1 gene deals with breast cancer. 80% of the gene, I'm sorry, 80% of the people who have this mutated BRCA1 gene will develop that breast cancer. So not everyone will develop it. Pleiotropy, just because you have the genes for it doesn't mean you're going to express it. And this is just having more than one digit or more than five digits. I love putting this in here because again, you can have twins that with the same exact genetics, but one can roll their tongue and one can't. And yes, rolling your tongue is genetic, and there's a little bit known like it is genetic, but it's also environmental as well. It's very interesting. Pleiotropy. Oh, I'm sorry, that wasn't pleiotropy. That was polydactyly before with the multiple digits on the hand. This is when one gene controls more than one trait. So you can see this strain has many different features that are because of this just one gene. So like people with albino skin or albino features, they have one pigment gene that's misfunctioning, and it's going to create several phenotypical changes. And so they have like lighter skin, lighter hair, lighter eyes. It's because one gene is controlling all of those. People with dwarfism have a mutation in one of their, I think, their growth proteins. I forget what specific protein, but obviously they have many different phenotypical differences because of that, just that one gene. Epistasis, this is where we have one gene controlling another, and you can see here you control gene A controls gene B. So if gene A is activated, that means gene B can be activated. But if gene A isn't activated, gene B, it doesn't matter if it's activated or not. Gene A is controlling it, so it won't be expressed. Um, labs are this way, where you have this pigment gene and the quantity of the pigment gene, and you can see the if gene A is expressed, that means they're going to be black. If gene A is expressed, but we don't have gene B, you're going to have like these brown labs or the chocolate labs. And then if gene A is turned off, it doesn't matter what gene B is, they're going to be these yellow